Thanks, Brian. So as also the program director, I feel like I have more latitude than maybe all the speakers and the topics that I asked them to talk about, because I'm going to deviate even from my own topic for a couple of reasons. One is for the sake of time, and I'm going to either skip or blow through some of the slides that I might otherwise spend more time on. But the other and more compelling reason is someone, and I apologize I didn't catch your name, asked a question earlier about what causes dystonia. And I'm not going to answer that. I wish I could. Many of us wish we could. But I think it's something that is worth talking a little bit about. I'm going to touch on it for a few minutes, just off the cuff. But I'm also going to try to catch us up on time, because I want to make sure we have time to hear about Jin Young's phenomenal research work that's very relevant, and also some of the stuff we're wrapping up with at the end with Charlene, because I think it's very important too. So I'm going to be brief, but I'm also informal. So if you guys want to interrupt me, you can, but I'll take the discretion of saying whether or not I want to take any time to answer your question. I'll be very brief in my reply. Okay, so let's roll. So let's dissect this a little bit. What do I mean by outcome measures? Well, I think everyone knows what a measure is. We're measuring something like with a ruler or what have you. Outcomes, the word is important because you want to know, for example, if you're given a treatment, what's the outcome, right? It's a very simple notion, but the idea simply is you want to measure severity before and after, and hopefully the severity is less afterwards. Again, really simple, right? But much of research, clinical research, especially clinical trials, hinges on this, and it's just so critically important. Even though this aspect of it isn't necessarily going to tell us about what causes dystonia or the mechanisms, but it's important for getting new treatments out there that are not just, ad hoc things, but are FDA approved and ideally something that insurance will cover ultimately. Okay. So, there's this simple notion, before and after the intervention. And these interventions, I use the term broadly because it can refer to oral medications, botulinum toxin, deep brain stimulation, TMS, TES, TDACS, physical therapy, placebos. What if we wanted to see if a placebo worked? We'd measure severity before and after, right? It's a very general purpose. And there's a lot of ways of measuring severity. You can ask the doctor. And this is the traditional way it's done in clinical trials, where the doctors have these clinical rating scales. Brian touched on one of them earlier, where they rate you on a scale, typically say 0 to 4. Say 0 is you're fine and 4 is you're really bad. And they look at you and they say, yeah, I think you're a 3. And you write that down. And then we do the same assessment after the trial of whatever the intervention is, and they look at you and they say, yeah, I think you're a 2. Right? And those are clinical rating scales, and there's a lot more nuance to it than that. I don't mean to be flippant about it, but that's the gist of it. Then, of course, and this is paramount, why don't we ask the patient? And actually, this is something that's gotten more attention in the past 10 years or more. The FDA has had an increased emphasis on what does the patient think? How much is this really helping? That's really what the whole enterprise is all about, after all, right? And that goes by a couple different terms, either patient-centered outcomes or patient-reported outcomes, because the patient is reporting it. Really simple, right? And then the third category, and I call these objective measures, and that is, ask the technology, the computer, or the device that's measuring you, or the, these days there's a lot of capabilities on smartphones in our hands, and all the hype about AI. It's not always wrong. It's got some potential. So let's dive into these a little bit. I'll be very brief. Here's an example, and I think this has been mentioned in at least one, if not a couple of the prior talks. One of the classic scales for dystonia, named after a couple of investigators, Burke, Fawn, and Marsden. It's the BFM scale. And the details here are not that important, but it's a common one, and it's got elements for each of the different body regions, including the eyes, which of course are relevant for blepharospasm. And there's two components, a severity component and a provoking component. Now I point this out because this is interesting. The provoking one is kind of like what kind of actions or how often is the symptom present? Or what kind of actions instigate it? Right, because we all know how dynamic this is. You might have blepharospasm, and then when you're talking, your symptoms are gone. Right, so it depends on what's happening. But then also there's the severity factor, right? And this is an interesting one. If you look at this, it's got things like exaggerated blinking, spasms, and then spasms with the eyes closed greater than a certain percentage of the time. Well, what if someone has exaggerated blinking and spasms? What number do you give them? It's not immediately clear, right? Even a simple ordinal scale that only has 0 to 4 as options. And this is just one example. There's a ton of examples. I could spend hours, if we're gonna write a review paper on this, criticizing all these different, there's like 5 or 6 different scales that have been used for blepharospasm that all have these kind of design deficiencies. And these lead to all kinds of problems. Go ahead.
With the design deficiency or kind of just something that is a truism is that we're all on different like 12-week scenarios, and so the plateau and the diamond line, whatever you want to call it, you're basically all on different scenarios.
Yeah.
And with injections, do injections work? Do they not?
So I'm gonna cut you guys off sometimes, and I apologize, but I want to catch your question. So the question is, and this is so variable across different patients in terms of the intervals between your injections and whether they're effective, et cetera. That's another degree of complexity. Actually, I'll talk about in a subsequent slide. You might recognize this guy here, but also I think he also put up a picture of one of our close collaborators, Sarah Perrier-Richardson, out in New Mexico in Albuquerque. They have spearheaded, especially Sarah and other people in the Dystonia Coalition like Buzz Jenna, have spearheaded this effort to develop a new patient-reported outcome that's undergone an extensive formal process for development. And the whole art and science of developing these scales has actually improved over the years. And the good news is there's a lot of excitement about this. There was a rigorous process that went into this, and this was just published, I think, a month or 2 ago now, right, Brian? So it just came out. And I'll just give you the tip of the iceberg. It's got questions about several different things. Of course, there's the motor features of blepharospasm, disability, kind of impacts on daily living, and then also psychosocial impacts. And there's several questions under each of these categories that have been really well vetted with input, not just from the doctors, but also patients. Maybe some of you here participated in this. In fact, I think Charlene and Jane were co-authors on this paper, if I remember correctly. So you guys must have been involved. Maybe more than you wanted to. But the cool thing is not only did they come up with this new scale, but also they have developed a pilot version of a cell phone app in which you can answer these questions in the app at home. It's not just something you do when you go to the clinic to visit the doctor. So you can, on a scale of 0 to 10, I don't expect you to see it here, but you can rate this on a weekly basis, for example. Is what was done in the current study, right? But it could be done more frequently. This is important, right? Because otherwise you go to see the doctor every 3 months and they ask you, how was it? Well, you got to remember that whole 3 months, right? We've got computers for this. We don't need to be doing this as humans. Okay. Now, here's the problem with whether it's asking clinicians for their impression or patients for their own impression. We're all humans, even the doctors. And so these are inherently subjective human judgment. And I'm pausing for effect, right? Because sometimes these things are referred to as, well, the patient's assessments are subjective, but the doctors are objective. I mean, I've seen this in published literature. No, they're human judgments, they're subjective, okay? And it's gonna be affected naturally. This is not a knock, this is just the way things are, right? It's naturally affected by whatever your training was, where you did your training as a neurologist, right? What your experience is. If you see Parkinson's patients all the time and you see one dystonia patient a year, you're gonna have a different assessment than if you see dystonia patients on a weekly basis, right? And so it's not that these assessments are wrong per se, but one of the biggest upshots of this is that they're variable. If I show you a video of a patient, 10-second video, and I ask you to rate it, and then I come back a month later and ask you to rate it again, you might not give me the same answer. If I take that video and I show it to 10 different doctors, I probably won't get 10 of the exact same answers. It's variable, right? Okay, so this has consequences. So you've seen already, I think in at least Brian's presentation, maybe others, some of these research studies where they show the outcomes of the intervention, DBS or any of the non-invasive ones. Well, how did patients do? It's really variable, right? There's treatment outcome variability. Some did really well, some got 80 or 90% improvement, some maybe only 10%. So there's a lot of variability inherent in the treatment across different patients. But when you add to that variability in the way we're measuring it, you've got 2 sources of variability. And so in clinical trials, the big hurdle they're facing, and the reason why it costs $1 billion to bring a drug to market, a big part of the reason is because of this box right here, measured treatment outcome variability. You've got 2 things contributing to that, right? So that variability, it's a long story, but in essence, it decreases the power your ability to detect effects, and that requires a higher number of patients, higher Ns, and that's going to require a longer duration of a trial, increase the costs, and it increases the business risk. Why are we talking about business in the blepharospasm meeting? Because we want better treatments, and a hurdle to better treatments is more trials, and drug companies aren't going to do it out of their own goodwill. They're going to do it to make money. That's capitalism, right? That's natural. It's a benefit of capitalism. But the challenge is they need to make a business decision, and if the costs are a lot higher, you need a lot more revenue to make it viable, right? For rare disorders, that's a tall order. So if we can push down the costs, we have a better shot because we've got a lower barrier to entry of companies going over the hurdle and committing the investment to try new treatments, right? Real simple. It's not rocket science. So the question is, what can we do with that measurement and variability? Can we get rid of it or decrease it? Well, this is the whole point of objective measures. If we can get past subjective measures that have all that variability, and we have objective measures that don't have that intrinsic variability, then the measured treatment outcome variability only comes from the treatment variability. Make sense? Okay. So this is really what motivates what we're doing. You got to always iterate between what are we doing and why, right? That's the why. So what we're doing is working on objective measures for dystonia. There's a number of groups that are. We're in a kind of golden age of, this has been around for a long time, but bringing engineers and scientists together with medicine to try to improve medicine. And in the space relevant for blepharospasm, there's two main approaches. Either EMG, the electromyogram, where we're measuring muscle activity, and you've seen a couple instances of that already, and Jin Young will talk about that briefly, a version of it. But then also you can do video recordings. And I think you've seen a couple images already that depict the complexity of the musculature of the human face. And this is one of the things that makes us actually uniquely human if you compare it with primates, or certainly non-primate mammals. Our facial expressivity is phenomenal. It's an important part of our nonverbal communication, right? It's got all these rich benefits. But it's probably also part of the reason why humans develop things like cranial dystonias, because of the rich complexity of that and all the neural machinery behind it. So, you're going to have a quiz at the end of the day, kind of a classic cliche on all these muscles here. The takeaway, it's complex. So in the EMG space, there's a great effort by a group in Atlanta at Georgia Tech that's also working with our collaborator Buzz Jin at Emory to try to develop some EMG methods that don't require the electrodes to be wired to an amplifier. They're wireless electrodes just connecting by Bluetooth, because of the advances in microelectronics and the nice things they can do with these really thin membranes and built-in electronics on the skin. But it's great because they've shown that if you compare an expert's assessment of whether, say, a video of a recording of the patient is a bleph patient or normal patient, to what's predicted by the machine learning algorithm that it's taking the EMG, the muscle activity signals, as input, and just an algorithm that assesses those, there's a reasonable degree of accuracy, of agreement on this. So it's very encouraging that this is something that could be used actually for diagnostics. Do you have bleph or not? Even though I'm talking mostly about measuring severity and quantifying it. But I want to point that out. But our focus has been on video, and there's a couple of reasons. So it's everywhere in movement disorders clinics. If you've ever been to a movement disorders meeting, kind of the lingua franca, the main thing everyone uses for their communication is depicting videos of patients, with their consent, of course. But it's a very visual disorder, right? People are mostly assessed visually. And you guys have probably all witnessed that, right? But EMG, there's some equipment involved, and in some cases expertise can be an issue, and of course the time to set things up. Whereas we can record a video. A lot of movement disorders clinics have video cameras at their immediate disposal, and we've all got one in our pocket these days. You can take a video and a snap without any additional hardware required or expertise. But also importantly, especially for dystonias, including bleph, it's less obtrusive. Many of you have sensory tricks. If you touch your face or something like that, sometimes you're physically holding up eyebrows or whatever, right? That'll affect your symptoms, desirably on purpose to try to ameliorate the symptoms. Well, we don't want our measurement methods affecting the thing we're trying to measure, right? Then you get what's called the observer effect. So at least with video, it's a little bit less obtrusive. So that's very important for measuring severity in the dystonias, including BLEF. Another very important pragmatic thing is, if we can measure severity with videos, then it really opens the door for all kinds of stuff, doing things at home, remotely, doesn't have to be at home, at workplace, whatever. We could assess things more frequently in different settings, activities of daily living, not just when you come into the clinic, right? Okay, so we've done some work on this. It started over about 10 years ago now. In fact, it started with Mark Hallett back in the early 2010s. And I'm going to blow through some of this stuff just so I can get to the other topics I think are more interesting. But basically, we can assess eye closure over time with great precision just based on the videos. And it shows agreement with some of the clinical rating scales. Okay, so it kind of shows convergent clinical validity. And we've done similar assessments in cervical dystonia. I'm going to blow by today for the sake of time. But I think an important thing too is to keep in mind when we're doing these assessments, it matters not only what the state the patient is, how long it's been since they had their last injection, et cetera, but also what the specific task is during the exam protocol. So there's kind of like the complexity of the parameters and the space for DBS and the TMS, et cetera. When you're doing assessments, there's a lot of potential variables in the experiments that need to be carefully controlled, right? Was the patient talking then or not? Is someone talking to them, or are they kind of in listening mode? What's the lighting like in the place where you're recording the video? Clinical settings, you guys can appreciate, are notoriously horrible for lighting, right, because of fluorescent lights. So these are just things one has to be careful about in terms of the research. I'm going to skip this for sake of time. I'm going to point out in the blepharospasm space, that earlier thing I showed was simply based on amount of time that the eyes were closed, a really crude measure. We're in the process of trying to develop more exquisite, fine-tuned measures for blinks specifically and spasms and different types of spasms, as well as the apraxia of lid opening we've talked about a few times. And this involves a lot of effort and some fine-grained analysis to kind of develop a ground truth with which to train up the AI system. So what you want to do is you want to go through the videos and ideally label some of these phenomena with very great temporal precision on the order of seconds or fractions of seconds. So I've had the good graces to have a great cohort of undergraduate volunteers at UCSD that have helped out with this the past several years, including some that are here today that I want to call them out. Come on, guys, stand up. I want to embarrass them. This is a great opportunity to embarrass them. So Vincent and Callie and Julianne, stand up for a second. Let me introduce you. So it's nice to have some younger people in the room. That's always a general thing, right? But the reason we really care about that is, I don't think that this disorder is going away anytime soon. And you always want to have the long view. And it's not only about helping us, but the future generations that might suffer from this disorder, right? And the causes are complex. We're nowhere near figuring those out. So it's really important and valuable to train up the next generation of people to get inspired and excited about this and informed about it. And that's an important part of the mission, right? So I'm really grateful for you guys and your help and contributions. And in many regards, we're just getting started, but they are key behind this enterprise. So when you support universities, a big part of it is, planting the seed corn, right? Okay. Yes. Ultimately, there's no silver bullet, even in the assessment space, just like in the treatment space. So the ultimate kind of combination will probably come from, a conjunction of, video-based measures, we call it our software CMOR, patient input, as well as the doctor's assessments. Because, the doctors have an exquisite eye on this thing. I don't mean to just relegate their opinions to the side, because they have exquisite expertise they bring to this, and that's cultivated over many years of training and experience. So I think the ultimate assessment will really come from combinations of these things. And we're currently engaged in a project that will look at the synergy between patient reports and CMOR, because patients can report on symptoms. That's the definition of symptom. It's something that you complain about, right, that you are aware of. as well as signs. And of course, there's a lot of overlap there, but they're not entirely identical. So if we have both patient input and objective measures, that combination can probably give us one of the most complete assessments of your severity. So that's kind of where we're going. And also, I'm going to mention Amy Lynn here because we're one of the studies on this that's relevant more for cervical dystonia, but also for BLEF. Amy's played an important role, and even though she's not here right now and I can't embarrass her anymore, at least you all got to hear from her before. So I'm really grateful for her contribution and her interest in dystonia. It's hard to get movement disorders neurologists interested in dystonia because all the effort's going into Parkinson's disease. It's just the way it is, right? But Raphael Pimenchuan, who isn't here, but Vincent, who you just met, are playing a key role in this study. And basically what we're doing is, for the first time, looking at videos midpoint between the toxin injections. So most studies in the past, they only recorded videos in the clinic when you came in, which is normally at the end of the 3-month cycle and when you're kind of putatively at your worst, right? Kind of a trough. But we want to see what the peak effect is in the midpoint. We've got that data and we're starting to analyze it. So it's very exciting. This is some of the first times we can get patient assessments and doctor assessments and objective assessments of what's the benefit of toxin injections in the middle of the cycle. Very exciting. I'm going to skip this. Okay, and this is the usual thank you. There's a ton of people that have been behind this. I've only mentioned a few. I want to highlight this column on your right. This is sponsors. There's often the question of, what can we do as patients and as society in general? And funding for research is key. And Brian brought this up before, and there's the question of what causes dystonia. The brain is incredibly complex, 100 billion neurons. Each one's got an average of 10,000 connections. And there's a ton of complex molecular machinery for each of those. So it doesn't take a mathematician to appreciate the brain is incredibly complex. Figuring out what's going wrong in blepharospasm is a huge, tall order, as with most neurologic disorders. And it takes a vast, difficult, protracted research enterprise and a lot of smart people putting a lot of effort into that to try to make progress on that front. It takes resources, it takes people, and it takes money. And anything you can do to help promote funding, whether it's through the BEBRF or at the federal level, when those things are under threat, that's chewing through seed corn. We're going to pay in spades for this for decades if the federal climate for funding is not improved. And I don't want to go into the political fray, but if we care about solving brain problems, we need to invest in that. And it's a long, difficult haul, but we have people that can do that stuff if they can do that instead of having to go find a job at Meta, right? So this, we've had the good fortune to have some federal funding before, but for myself and many of our colleagues, that's not a really rosy future. And I've seen a lot of brain drain. A lot of my good colleagues go off to the private sector and high tech and do cloud computing and AI and all that stuff to make better versions of GPT. That's great. It's not going to fix blepharospasm. So, when you talk to your congresspeople or you donate your $5 or whatever it is to BEBRF, that is so meaningful, as well as participating in the trials, right? I want to comment a little bit more about the causes concept, because I think about this a lot. I think, as with many complex brain disorders, partly because it's so complex, it's probably multifactorial. I think that was mentioned before. There's many factors that play into it. Those probably differ from one person to the next. But a couple things stem from that. One is, okay, if it's multifactorial in the causes, then it's just intuitive that the optimal treatments might be multifactorial. Right, so historically, let's take even physical therapy type of modalities, for example. Usually that's done by a physical therapist in complete isolation from a neurologist who might be prescribing different medications or treating you with Botox. Two different people, you get referrals to each, different offices, different institutions almost, right, usually. There's a theory that, at least for some of the especially task-specific dystonias, and musicians' dystonia is the extreme example, if it's something that's really subtle, that's abnormal in the biology, but a certain motor program is used repetitively, that only that combination leads to the manifestation of musicians' dystonia. I know BLEF is a different matter, but the idea is that Okay, if it takes both of those to get there, maybe we can modulate both of those to get out of it. We should be doing pharmacologic modulation as well as the PT type of things in very carefully coordinated synchronous fashions that are just not currently done. To be more specific about BLEF, if we think things like dry eye are a contributing factor, right, then modulating dry eye in a very carefully synchronized fashion with the timing and the doses and patterns of toxin injections would probably make a lot of sense, right? Those are things that could be done soon without a whole bunch of additional research. I could go on and on about this, but the key thing is we need to understand the brain circuits better. And our current brain imaging modalities like fMRI and PET, it's kind of like if you want to understand a computer, if you stick a stethoscope up to the computer and you listen. Everyone understand the metaphor? You know, when you've got 100 billion neurons communicating through 10,000 synapses each and a bunch of complex molecular machinery for each of those, you're not going to understand how the brain works through fMRI. Never. It's better than nothing, but we're never going to get there. Now, we also can't put 1,000 or 1,000,000 or 1,000,000,000 electrodes in your head either, not currently, not very easily, right? And we normally don't do that unless we're going in to intervene, like with DBS. The fact that we can do recording with DBS now is very exciting. That will give us some inroads to some insights to be able to record live in the human brain, right? But there are ways to develop reasonable animal models of dystonia that are where we've barely scratched the surface. So when I say animal models, I mean things in animals that kind of mimic, at least in some regard, some aspects of dystonias, including blepharospasm. Those things just don't get enough funding. But there's a lot of potential there to try to figure out what's going on in those brain circuits, because there's a lot more similarity between animal brains and human brains than most people appreciate. You probably heard the things like, oh, the genetics are like 99.9% the same. That's true. It's not quite that when you get to the circuit level, but it's pretty high still, and we can learn a ton from animal models. So those are kind of my sales pitches for research. I just want to dive into those for a few minutes since the question came up. We can't answer the question. I want to answer the question. A lot of other people do. And a lot of people have ideas about how to do it. They just need resources. Okay, I want to take questions, and let's limit it to just a few so that we can go to Jin Young. And I apologize ahead in advance if I'm too terse. Yeah, go ahead.
Is research being done on genetics as the factor? You have one family member with Parkinson's, another
Yeah. The question is research being done on genetics in terms of if you've got a family where one member has Parkinson's, one has cervical dystonia, one has BLEF? The answer is yes. As I think Brian alluded to earlier, that's a very active area of research. And in some ways, even though people think it's complex, genetics is actually way simpler than brain circuitry. So that's part of why we're making good headway in that regard. Having said that, for most adult onset focal dystonias, including BLEF, there still are no genes identified yet. They might be. There's open road in that regard. But if it's a truly complex multifactorial thing, maybe it involves 2 or 3 or 4 genes and environmental triggers. And so any one gene alone may not explain it. Only certain genes and certain combinations and combined with certain environmental factors will trigger it. That makes finding the genes much, much harder. Compared to Huntington's disease, it's so straightforward. It's one gene, however many expansions you've got of the CAG repeat, so much simpler. It's like the opposite extreme. Does that answer your question?
How do you find the researchers who are looking into this?
How do you find researchers that are looking into the genetics? Brian, maybe you want to weigh in on that? Do you want to?
I would say with the Dystonia Coalition, which unfortunately runs out of funding this year, not that it runs out of funding, it's no longer eligible for funding. It's ran 15 years. And, but in that study, we still have a very large database of genetic samples. And so when you do genetics, the tests might be easy, but you need thousands of samples to look for what's wrong in there. And so there's probably going to be a variety of studies coming up that still use that database, and to be able to to contribute to it, I'm not exactly sure, but what we do recommend typically is looking at clinicaltrials.gov and look under blepharospasm, and then you can find any federally funded research across the country that involves blepharospasm, and so that might be one eligible way to do, one way to do it as well. And I imagine the BEBRF newsletter can also reveal that when studies come up.
Yeah, good. Next. Heidi.
There's a lot of money going into Parkinson's. Is any of that research could help Parkinson's, or the research so different than it?
Great question. So if there's a lot of money going into Parkinson's research, how much could it help us, dystonia and bleph? There are little areas of overlap. I think they might have been mentioned earlier today. Some forms of Parkinson's can exhibit features of, or the symptoms of blepharospasm. So in that regard, some of it overlaps. There's also some interest in the possibility that in some cases, people can exhibit what looks like dystonia, and then it converts over time to look more like Parkinsonism. In fact, there's a specific, very specific genetically identified or associated form of dystonia called XDP, X-linked dystonia Parkinsonism, that does exactly that. So it's a little bit on the fringes, but there is some overlap. Yeah, if that answers your question. Yeah.
So Parkinson's has made a lot of progress since it became famous, because it had a famous spokesperson who was willing to become public.
We need fame.
Right, so I'm in another organization, another condition that I work with, where the newest person that self-identified publicly is Melissa Gilbert, who is in Little House on the Prairie.
Yeah.
And, you know, all of a sudden, there's more attention. So how do we find somebody?
How do we find a celebrity with dystonia? Yeah. That's a great question. Yeah, even though it's less common, the laryngeal dystonias, sometimes because it may have a higher incidence in people that are public speakers or have more public-facing roles or talk a lot in their profession, that might be one pathway through which we could find a celebrity sponsor or celebrity advocate.